I've been with Anika for over 5.5 years now. Um, and sort of as the director of research and development, I lead all the new product development activities here at Anika and their OA, pain management, and regenerative solutions, hearing from doctors and patients about how our products are improving the quality of life for so many people around the world. Anika has been working with hyaluronic acid really since its inception. Anika was founded over 30 years ago now, actually as a company exclusively to make injectable HA products. And since then, Anika has become a global leader with a broad portfolio of dozens of HA-based medical devices and therapeutics. And also in that time, Anika's pioneered a number of new technologies, with over 50 hyaluronic acid-based patents granted worldwide, and helps tens of millions of patients. So Anika really has a lot of expertise in hyaluronic acid, its use in medicine and therapeutics, and how to modify hyaluronic acid for specific pathologies and applications. Where can Hyaff technology take us? So when I first joined Anika, I knew a little bit about hyaluronic acid, but I was really not familiar with the Hyaff technology. And the more I learned about it, and its use in tissue regeneration, cardiovascular, smooth muscle, tendon, cartilage, bone, the more I was in awe of the vast potential of this technology. I truly think that the sky is the limit for the Hyaff platform. Unlike other HA-based products, Hyaff can be made into a variety of solid forms, from fibers, textiles, nonwovens, films, granules. You can even make Hyaff into molded shapes in specific and complex geometries. So really, any application where doctors are currently using collagen or other tissue scaffold materials, for implants, scaffolds, tissue engineering, wound dressings, drug delivery systems, these could all be potentially replaced by Hyaff. Hyaluronic acid's form in the human body is, so hyaluronic acid, the molecule itself, is a polymer. It's a long linear chain molecule. In the body, it is in a gel form because it is extremely hydrophilic, so it absorbs a lot of water. So depending on the length of that chain, it can form a viscous liquid or a gel. And it's naturally occurring. It's present in all tissues of mammals and most vertebrates. And the body can readily metabolize and regenerate it every day. So it's found really anywhere in your body, from your gums, skin, hair follicles, connective tissues, joints, vitreous bodies. What hyaluronic acid does in the body can do a number of things, and a lot of it depends on the length of the chain or the molecular weight and the application that it's in or the anatomy that it's in. So in some aspects, like in joints, it provides viscosity. So visco supplementation for that joint is what the HA is doing. In other applications, like in dermal and skin, it's mostly providing hydration. So because of the hydrophilic nature of the molecule. In other applications, like for when you have an injury, there's a wound healing response where HA is used to do some cell signaling to initiate the wound healing response, as well as inflammation regulation during the wound healing process. So it really just depends on where it is in the body and what needs to be doing. And HA can do a number of different things. At the cellular level, HA interacts with cells in a number of ways. So first and foremost, it does provide a lot of hydration for the cells, which is critical for cell function. As I said, it provides a lot of cell signaling for directing the correct cells and cell pathways for wound healing. It also has a lot of cell receptors that are involved in inflammatory response regulation. So it does a number of things, and depending on the types of cells or the environment it's in, it's going to do different things as well. And certain cells that interact with HA, it's been shown to interact with a wide range of cells in the body, including mesenchymal stem cells, fibroblasts, chondrocytes, osteoblasts, as well as endothelial cells. And to your last point about what role does it play specifically in cell signaling and differentiation and the healing response? So, there are a few cell receptors that HA specifically interacts with. Two of the main ones are CD44 and RAM or HAMM. These interactions help to regulate the cellular adhesion and migration, as well as proliferation and modulation of the inflammation. And these are critical parts of all phases of the wound healing response, from the inflammatory response to proliferation to even the remodeling phase. You can sort of think of two different realms of healing. There's the embryonic healing, and then there's adult healing. So I guess first, if we start with adult healing, this involves a robust and prolonged inflammatory response, and this often leads to things like fibrosis and scar tissue. Now, for embryonic healing, like you said, it is in an HA-rich environment. So because of that, it has a more hydrated, low viscosity matrix that facilitates cell migration and morphogenesis. So this ends up resulting in suppressed inflammation, scarless healing, and tissue regeneration. I think introducing HA into the wound environment is providing more of an embryonic-like healing that's going to reduce scarring and improve that tissue regeneration, as opposed to just the fibrotic scar tissue. The human body, and in fact all mammals and most vertebrates, produce hyaluronic acid in their body, and they metabolize it as well. So in vivo, HA is produced by hyaluronin synthases, which are membrane-bound enzymes, and it's degraded by hyaluronidase. So for a typical young adult human, they have about 15 grams of hyaluronic acid in their body at any given time, and about a third of which is broken down and replenished daily. How does the body metabolize hyaluronic acid? You have these hyaluronidase enzymes that will break down that long polymer chain into essentially smaller chains. And then you'll have additional enzymes break down those smaller chains into essentially their small molecule forms. And those are then further metabolized by the body. And then ultimately the cycle is continued, and hyaluronin synthase will use those basic structures to recreate new hyaluronin in the body. How the body breaks down Hyaff compared to hyaluronic acid really comes down to how Hyaff and hyaluronic acid differ chemically. So Hyaff is an esterified form of hyaluronic acid. So basically you have the hyaluronic acid is reacted with benzyl alcohol, and you form benzyl ester groups on the backbone of the hyaluronic acid. So when it degrades, first you have to hydrolyze those ester groups. So you get the benzyl alcohol back as well as that original hyaluronic acid. And then the body will metabolize that hyaluronic acid by the same pathway that that unmodified hyaluronic acid will go through. So it's essentially an extra step that the body has to go through to metabolize. First, deesterifying the benzyl ester off of it, and then metabolizing the HA. The body can naturally metabolize benzyl alcohol and excrete it through urine, and it can also metabolize the hyaluronic acid component into its smaller parts and then reassemble them into new hyaluronic acid that the body will use. For Hyaff, yes, the way that it differs from the endogenous hyaluronic acid is that it is modified in a way that it becomes less hydrophilic and more hydrophobic. And so what this does is it slows the absorption of water. It takes longer for it to hydrolyze over time, and then it takes longer for it to degrade and resorb over time as well. So this greatly increases the residence time of the Hyaff material over our unmodified HA or even our cross-linked products. Yeah, so HA is going to have a little bit different response in vivo than a xenograft collagen material would. So, first off, xenografts, while they're very safe, there's always a small but inherent risk of animal pathogen transmission, or inducing an immune response, or triggering a reaction to individuals who are allergic to the given animal protein that it's derived from. But beyond that, we've found from our preclinical studies that while collagen xenografts do provide, a small amount of improvements to the healing response to soft tissue, the Hyaff materials have the potential for providing significantly more healing capacity in the same applications. We see a couple differences at different stages of the wound healing process. So, in both cases, you have a very high inflammatory response at the early stage of healing. And then when you get to sort of the longer, like 6 to 12 week time points, that's when you sort of start seeing a difference between the response of a collagen material to the response of the Hyaff material in the soft tissue models. And a lot of that comes through what some of the cell signaling is doing from the HA as it's being released. So there's a concept of macrophage polarization, where there are two types of macrophages. There are M1 macrophages, which are pro-inflammatory, and these are typically associated with necrosing tissues. And there's M2 macrophages, which are anti-inflammatory and are typically involved in regeneration or in repair of tissues. And we've seen that for our Hyaff implanted materials, we tend to see a lot of these M2 macrophages inducing a lot of healing of the tissue, as opposed to just removal of material and forming of scar tissue. In the collagen implanted animals, we saw significantly less of the M2 macrophages, and so the wound would heal, but it wouldn't necessarily regenerate new tissue. So why is HA so versatile in different applications in the body? HA does a number of different things for different parts of the body, from, as we said, the hydration or the lubrication or cell signaling, and inflammation modulation. So I guess I'll just say that generally speaking, HA plays a crucial role in all aspects of sort of the wound healing process. And if you look at specific applications, like take cartilage, for example, it's going to be really important for cell signaling for chondrocyte differentiation and stimulation of proteoglycans and glycosaminoglycans, which are components of healthy cartilage. And in addition to that, it provides viscosupplementation and lubrication to the joint space. So viscoelasticity and lubrication is going to end up protecting the joint, and that's going to in turn promote chondrogenesis. For tendons, it provides some viscoelastic lubrication and adhesion reduction. So there's some literature up there about a tendon gliding effect could reduce scar tissue. And HA also promotes enhanced tenocyte viability and collagen production, and has been shown to promote angiogenesis in tendons and muscle applications. Why does HA work in these different applications for these products? Part of the reason is because HA does so many different things in different applications, but the other part is because we actually tune our HA to be used in different applications. So, for example, for osteoarthritis, the main primary mechanism of action for a product there would be viscosupplementation. So we're going to want a high viscosity gel or a high viscosity fluid. So we use our either unmodified or cross-linked HA in a gel form. If we were to be making cosmetics, we'd want hydrophilic properties. So we'd want a unmodified HA that could swell and retain water. Similarly, for ophthalmic products, we would want something that has a really good lubricating effect, or as in used as a vitreous substitute. So we would tune our HA to mimic vitreous fluid rheological properties. And then for tendon healing, we ideally want something that can be made into a scaffold that will allow for cells to infiltrate and adhere to the scaffold. And then as that scaffold degrades, it's going to release HA, promote some cell signaling, and promote a healing response. So for the tendon healing products, we use our modified hyaluronic acid, once again, the Hyaff technology. So this provides that hydrophobic structure we need for cells to adhere and proliferate and migrate. And as that Hyaff degrades, the hyaluronic acid is released into the wound space to promote that new tissue growth that we need. So when we are designing a scaffold for our HA products, there are a couple key aspects that you need to make an ideal tissue scaffold. So first off, it needs to be a porous 3D structure to allow for cellular infiltration. So you can make this either through fibers or through some sort of phase induction process, or a knitting process, or a non-woven spinning process. And for Anika's products, we utilize two main technologies for our scaffold. So for Hyalofast, we have our non-woven Hyaff scaffold, which provides extremely high porosity, a lot of space for cells to infiltrate and attach. And then as the whole scaffold is degradable, tissue completely engulfs where that scaffold was. For the Integrity product, we have a knitted structure of 80% Hyaff by weight, as well as 20% of polyester PET by weight. And we create the Hyaff fibers by a wet spinning process in Bedford. And then we bundle those fibers together. We put them through a warp knitting process along with the PET to provide a high strength scaffold. So it's very thin, it's got high porosity, and it's got good strength for robust deployment in arthroscopically. The scaffold is also laser cut along the edges to prevent any fraying of that knit structure when deploying. So there's a number of cool technologies that we have employed for both our Hyalofast and our Integrity products. Three biggest aspects for use of our Hyaff scaffold in rotator cuffs are certainly going to be porosity is one of the biggest ones for cell infiltration. Second one is going to be sort of the density, or maybe you could even think of it as the dose of the Hyaff or hyaluronic acid into the space. And then the third thing is the high strength of that scaffold that allows it to be manipulated and deployed in situ to the wound site. So what does the body of literature say about hyaluronic acid and Hyaff clinically? I will say that HA has been used clinically for probably about 70 years now. Specifically in orthopedics, HA injections have been used on tendinopathies since probably the early 1980s, and there's several publications about that. A lot of these publications have proposed mechanisms of improved healing by reduction in the inflammatory response and the chronic inflammatory response, as well as improvements to tendon gliding and promotion of healing by cell signaling. For Hyaff, there's significantly less number of publications. There's was a lot of research on it in the early 2000s for use in tissue engineered vascular grafts. And then there were a number of publications after that when Hyaff was used a lot in some wound dressing applications, specifically Anika's Hyalamatrix product for dermal and epidermal substitutes. And it showed excellent wound healing results over a paraffin gauze control. And then even more recently, there was been some publications, 10 years ago on clinical use of Hyaff mesh on treatment of ruptured Achilles tendons with excellent outcomes. And now we're seeing a lot of good results with our Integrity and Hyalofast products as well. So when I talk to surgeons in our clinical labs, a lot of times the most common question is really how the Hyaff material differs from hyaluronic acid. I know we a lot of times market stuff like our Integrity and our Hyalofast products as HA based or HA scaffolds. Chemically, they are a little bit different. They're Hyaff, and a little chemical change to a molecule makes a big difference to the properties and how it behaves in the body. But the really cool thing about Hyaff that I always try to convey to the surgeons is that even though it doesn't behave at all like you think hyaluronic acid should behave, when it degrades, it becomes hyaluronic acid. And the other degradant, benzyl alcohol, is also a natural chemical that the body can readily metabolize. So the really cool thing about this, as opposed to other synthetic polymers, synthetic degradables like PLGA and PCL, the Hyaff material degrades into a natural polymer that the body knows how to handle and resorb and is actually beneficial to the healing, as opposed to some other of the synthetic degradable materials. From what I've seen from the literature of other treatments and from what I've seen with Integrity, if I had a rotator cuff repair, I think I would get the Integrity implant because the response that we've seen clinically has been great. And I know that without any augmentation, the chance of re-tear is incredibly high. And the last thing I'm going to want if I have a rotator cuff tear is to get a re-tear. So yes, if I got a tear right now, I would definitely get the Integrity implant.